Good evening, students. Hope doing all for examination, NEET examination. I think you are all well prepared throughout the year, and also repeaters also they are going to write examination. So this is not a time to study the new chapters and also new topics, but for interest of the students, just to have to recollect the botany, those the concepts which are difficulty area, I have to just recollect, that's only. So from the beginning we have to start from majority of the students they are asking, sir, newly added families, how can we are going to attempt those questions? But one thing, the NTA cannot able to going to difficult about the families, because it is newly added concept, so that only those areas which are necessary we have to just collect. So let us start with the newly added families. Newly added families. So already you have three families regarding family Fabaceae, Solanaceae and Liliaceae. Already you know, you did learn very clear about the three families. But at least the newly added families, I have to go with Asteraceae and also Poaceae and also Malvaceae. So from these three areas, what concept we have to learn, that is important. The first of all, the floral characters are very, very essential. That is, for example, when come to the Asteraceae members, mostly there are two types of the flowers. One is the ray florets and second one is the disc florets. From these two areas, they are asking ray florets are, they are the female flowers and zygomorphic flowers. So that's only they are asking. So ray florets are female flowers and zygomorphic. So from this area, if they are asking stamens are present, that is wrong answer. From disc florets, this is the bisexual flower and this is the actinomorphic flowers. But it is having the stamens number is 5 stamens and epipetalous condition. And when come to the gynoecium, it is a bicarpellary syncarpous inferior ovary. So from this area you have to remember that's only. So regarding flowers, only the Asteraceae members, they are having two kinds of the florets, not flowers. They are the florets. So that is the most important area you have to learn. And also the important, what kind of the inflorescence present in the Asteraceae, that is head inflorescence, that is the advanced type of the inflorescence when compared to all the inflorescence. And what kind of the fruit we have to observe in the family Asteraceae. I think you know the cypsela fruit. So that's you have to learn. And also the cypsela fruit, it is having a special character, that is pappus, having pappus. If the question is asking pappus is the parachute mechanism, sometimes they are asking parachute mechanism seen which of the following type of the families the question is asking, you go with the Asteraceae. So these kind of the questions you have to expecting from Asteraceae members. And also what kind of the stamens are having, syngenetious stamens and also epipetalous condition. So these two are the important here. So that's all about Asteraceae. But of course, if they are asking Asteraceae, economic importance, chrysanthemum and sunflower, these are the important. I think sunflower, you know, that is a oil yielding plant. The seeds are getting oil yielding plants. But of course, chrysanthemum, marigold, that is the ornamental plants. And also Parthenium hysterophorus. Parthenium, it is the pollen allergic plant also belonging to Asteraceae members. So that is mostly you have to remember. And when come to the Poaceae, if you go with the Poaceae, Poaceae also it is a newly added. This is the monocotyledon and this is the zygomorphic flowers, you can see. This is also bisexual flowers and zygomorphic flowers, you can see. This is not having calyx and corolla, only perianth is present. There is a two perianth that is formed as a laudicules. If they are asking androecium, 3 stamens are present and some are 6 also will be there like bamboo. And gynoecium, it is a monocarpellary syncarpous superior ovary and this is also basal placentation. Here also basal placentation. So these things you have to remember regarding Poaceae. In the Poaceae also you have to remember spike is the inflorescence. In the spikes of the spikelet you have to remember glooms, palea and lemma you have to remember. and also perianthropes you have to see, and the stamens, and also gynoecium is present. So those, and what kind of the fruit is present in the poaceae member? That is caryopsis. Caryopsis. The question is asking caryopsis. And also caryopsis is a monocot seed, so that this is endospermic seed. So caryopsis fruit is having endospermic seed, that is a triploid in condition also, you have to remember. And also economic importance, wheat, Oryza sativa, paddy, and also maize, these are all the cereals. And also bamboo is one of the important, sugar cane. So I think you have to remember regarding about all these poaceae member, economic importance also. And when come to the Malvaceae member, it is also newly added family. So Malvaceae, it is also called as show flower family, mellow family, we can see. And once you remember the plant, you can easily remember the characters also. For example, as you know, the best important plants are hibiscus. One is the hibiscus. And also, frequently I saw bendi, Abelmuscus esculentus, and also cotton. Bendi means lady's finger. If you see these three, immediately you can remember it is a valvate estuation. You can say in Malvaceae members. Valvate twisted estuation we can see here for Malvaceae, twisted estuation. And also the important thing is Malvaceae, it is also epipetalous condition. When come to the stamens, it is a monoadelphous condition, stamens, epipetalous condition. And gynoecium we can see pentacarpellary syncarpous superior ovary, basal placentation, axial placentation. This is the most of the Malvaceae members we can see. But if you come one by one, for example, epipetalous condition, and also the members of these Malvaceae members, the fruit is important. That is, capsule is present here, capsule. So which of the following families are having capsules? You can remember Malvaceae member, Solanaceae member, and also Liliaceae. But regarding asking the question, you have to just change it. Most of the capsules, some are berries. While the question is asking, you can go with Solanaceae, Liliaceae, and vice versa. So mostly, do not worry about the newly added families. First of all, you have to remember carefully. The floral characters, when you remember carefully, you can easily getting the answer. And I think I can expecting mostly match the following, you can getting the questions from newly added families. Of course, it is also actinomorphic flowers, these are also, and bisexual flowers we can see. And one more important area, of course, the morphology, I think you can easily attempt the questions. So, like the areas of estuations, like the areas of placentations, and like the areas of leaf, you have to observe carefully. While you are attempting the answer, first you read carefully the question is asking. Sometimes they are asking petiole. General functions of the petiole is different, and long flexible petiole function is different. So that kind of the questions you have to remember. And also, if you go with the flower, carefully you can read the flower, what kind of the flower they are asking. For example, asymmetric flower canna, symmetric flowers, datura, brinjal, these are all the example. If you go with the zygomorphic flowers, all Fabaceae members are zygomorphic flowers. So like that you have to attempt the questions. Okay. When come to the recently I am observing some questions are asking in molecular biology, some tricky areas. Of course, the students are frequently asking the questions. For example, mostly recently I am getting one question. Of course, one of the major institutions are asking the question. For example, if the number of the chromosomes present in G1 of the human cell, is given something. Human G1, if their question is asking, you can go with the number of chromosomes or 46 chromosomes. But they are asking what is the number of chromatids present at metaphase they are asking. Metaphase. If the medium is having radioisotopic thymidine. The majority of the students did mistake from this area, but I will give the clarity. First of all, in G1 stage, how many chromosomes ratio between the chromosome and chromatid? One chromosome with one chromatid. Chromatid in the sense it is DNA content is having one DNA. But when go for S phase, prophase, and S phase, G2 phase, and prophase and metaphase. What happens here? When come to the S phase, what happens? The DNA content doubles. So these DNA, it will take the thymine from this area, so that automatically by the semi-conservative mechanism, what is happening? One radioactive thymidine, it is having two DNAs and it is normal DNA. So if you go with the metaphase, what is happening? It is a 46 chromosome into 2 DNA, that is 92 chromatids are nothing but 92 DNAs are present. Of course, even though it is having one thymine strand, that will come the 92 DNA having the radioactive thymine. That is the answer. So like this you have to attempt the questions and answers. And also I can see some of the questions are asking in molecular biology and molecular basis of inheritance. They are giving the nitrogen base sequence on mRNA and they are asking how many maximum number of amino acids are getting from this mRNA. So if they are giving the data mRNA, simply what is the polarity, that is important. The polarity in the sense 5' to 3', if they are given, that is already it is a mRNA. So in this nitrogen frequency, already they are giving the AUG, that is the start codon, and you can see where the stop codon is observing, in between how many codons are there, that is indicated the number of amino acids. So that is the question. But frequently what they are asking, there is a newly added nitrogen base or nitrogen basis is deleted. If they are asking the question, you can just delete the particular area, and where stop codon is observing, from in between only you have to take the number of amino acids. So that is most important. If any added the nitrogen base or delete the nitrogen base, and you can count the total number of nitrogen bases and divide it into 3 codons, and you can just make it into number of amino acids. Where the stop codon is mutant, that is very important. Okay, so these kind of the questions also I am observing. And also from this mRNA, how many amino acids will be obtaining? If they are giving the number of amino acids and they are asking the number of nitrogen bases, and also codons also they are asking. So like this you have to remember carefully. And this area, and most of the students are asking some area like respiration asking. So I will just briefly discuss about the respiration and where you are committing mistakes. I will come to that area. For example, in respiration, one thing you have to remember. First of all, if the question is asking as a glucose is a respiratory substrate, you can go. The total ATP is 40. Net ATP you can just 2 ATP will be removed, and the answer is 38 is the net ATP for complete oxidation of the glucose. First you have to remember this area. And second most is, what is the net ATP for fermentation, whatever they are asking, fermentation. Whatever they are asking, lactic acid fermentation and alcoholic fermentation, whatever they are asking, the net ATP for fermentation is 2. These two are coming from glycolysis. And some students are getting confusion, sir, of course in the fermentation NADH2 also formed, but that is used in the fermentation, so that how many number of ATPs are loosed in fermentation. If the question asking 6 ATPs are loosed, because they are used that one. So of course that is other question. Glucose, if it is a respiratory substrate, total ATP 40, net ATP is 38. Fermentation, whatever the fermentation is okay. But sometimes they are asking in link reaction, separately they are asking, it is a gateway of pyruvic acid, oxidative decarboxylation of pyruvic acid in link reaction. So there are 2 NADH2 into 3 ATP. So that is 6 ATP. That is 6 ATP. You can see here, For link reaction, you can get 6 ATP. That's it only. But majority of the students are getting confusion, whereas in Krebs cycle. Dear student, just remember what the question is asking in the Krebs cycle. You have to observe carefully. In Krebs cycle, how many total number of ATPs are formed? By one glucose complete oxidation, you can go with the 24. That is one. But if they are asking one Krebs cycle, you can go 12 ATP only. 12 ATP. For glucose complete oxidation, glucose, what is the direct ATPs in Krebs cycle? 2 ATP. For one Krebs cycle, they are asking 1 ATP. So only for oxidations, how many ATPs are formed through Krebs cycle for complete oxidation of glucose? So you can just subtract 2, 22 is the answer only for oxidations, for glucose oxidation. For one cycle they are asking 11. So for this area you have to remember when 24 is coming, when you have to write the 22, when you have to write the 12, when you have to write the 11, when you have to write the 2, and when you have to write the 1. There are majority of the questions are asking. But sometimes they are asking, if the acetyl coenzyme A is the respiratory substrate, you can write 1 Krebs cycle. If the pyruvic acid is the respiratory substrate, you have to take the link reaction also you have to take. So that is very important. And in respiration, you can remember SLP reactions, decarboxylations reaction. You can remember the students in glycolysis, neither CO2 is releasing nor oxygen is releasing. In link reaction, one step CO2 is releasing. In Krebs cycle, there is 2 steps CO2 is releasing. And total number of oxidations in complete aerobic respiration, glycolysis 1, link reaction 1, and Krebs cycle 4, totally 6 oxidation has been taking place. And total decarboxylation steps means 1 in link reaction, 2 in Krebs cycle. There are 3 steps. So decarboxylation is only takes place in mitochondrial matrix, not in the cytoplasm. And these are the majority of the questions. And not only these areas, and I think students are learned all the areas from the Krebs cycle. And RQ value you have to remember carefully, and what kind of RQ value they are asking. For proteins 0.9, for tripometic acid 0.7, for carbohydrates like glucose is a unit, that is 1, and whereas in succulent plants 0, anaerobic respiration is infinity. And based on this, sometimes they are asking arrange the questions like this. So this is the area from respiration, and intro points you have to remember. And NADH2 is slowly oxidized in fermentation and vigorously oxidized in aerobic respiration. That area you have to remember. And glycolysis also you have to remember. 2 redox equivalents are removed from which of the following compound? It is a G3P, triose phosphate. So that is a 2 redox equivalent. Sometimes they are asking. So this is the areas mostly you have to remember. Right. And one more very important the area students are asking, plant kingdom. So of course it is also, when compared to all the areas, the plant kingdom also the tricky areas. First of all, the students are remember that which is the gametophytic stage, which is the sporophytic stage, which is the dominant, which is the independent, which is the independent. That's you have to remember. One thing I have to just asking here. First you have to divide the total plant groups. One should be algae and bryophytes, pteridophytes, gymnosperms, and lastly angiosperms. Of course the angiosperms, even though deleted, but of course consider the angiospermic nature, it is relevant to the gymnospermic nature. So first of all, these two are non-vascular. From here, these 3 are the vascular. These 2 are the flowering plants. These 3 are the non-flowering plants. So I am just explaining briefly only, not detailedly, but these areas are very essential for you. So one thing, embryo absent. embryo present, embryo present, embryo present, embryo present. If the question is asking zygote, zygote is present in all sexually reproducing organisms. Algae, gametophyte is dominant, whereas the bryophyte, gametophyte is dominant. Here the sporophyte is depend on the gametophyte, but the pteridophyte, sporophyte is dominant and independent. Here gametophyte also independent. In gymnosperms, the sporophyte is independent, also dominant. And the students are remember that the gametophyte in gymnosperms is highly reduced, and frequently the question is asking from this area. Which of the following plant groups are having highly reduced gametophyte? You can come gymnosperms and angiosperms. And also frequently I am just getting known about which of the following plant groups are having a very least number of gametes. Also you can come gymnosperms and angiosperms. Least number of gametes in gametophyte. For example, you can take pollen grain, it is having least number of gametes. But if you take pteridophyta, gametophyte is independent, so that more gametes will be releasing. Here also more number of gametes releasing. Here also more number of gametes are releasing. And so on the same what the characters are given here, gymnosperms, the same should be generalized for the angiosperms. It is the sporophyte is dominant and independent. Gametophyte is highly reduced on the angiosperms. So that is very important area, angiosperms. And this is the question. But if you take all the members, gametophyte is non-vascular. Remember the student, gametophyte is non-vascular in all the members, even though gymnosperms and angiosperms. So from this area, the students are getting some confusion, so that I have to remember that algae, bryophyte, pteridophyte, gymnosperms, angiosperms, dependent, independent, which is diploid, which is haploid. Of course, wherever you find the gametophyte is haploid, wherever you find sporophyte is diploid. So this area you have to remember. And also there is no vascular system, so that you need not write xylem, phloem from this area. From here onwards it is a vascular. But here you have to remember the students, xylem, it is having only tracheids, no vessels. Here the xylem also having only tracheids, no vessels. But here the xylem is having tracheids and vessels also. And when come to the phloem, I think you remember that the gymnosperm's phloem is having sieve cells and albuminous cells. But whereas if you come to the angiosperms, the phloem is having sieve tube members and companion cells. So that is the area. And also when come to the pteridophyte, Selaginella and Salvinia, they are the heterosporous nature, precursor of seed habit but not seed producing. Remember the student, very important question. The pteridophyte is not seed bearing plants, but it is a precursor of seed habit. But the seed bearing plants are coming from gymnosperms and angiosperms. So that all heterosporous, these two all heterosporous are seed bearing plants. Since these are the heterosporous, the gametophytes are always dioecious. So that these kind of questions, which is the dioecious, which is the monoecious, and you can remember. And also in bryophyte, you have to remember liverworts and mosses. Mosses are advanced sporophyte. Liverworts are somehow it is a primitive sporophyte. Why? Because the spore dispersal nature is somehow not advanced, whereas in the bryophytes of the liverworts, Markantia, it is a dioecious. And also rhizoids, you have to remember that is unicellular and multicellulars. And what is the sphagnum? You have to remember the students. Selaginella salvinia. And pteridophyte prothallus, it is grown in the cool, damp and shady areas. It is in most water is essential, so that the geographical nature of the pteridophytes are very less distribution. Majority of the times asking the question assertion and reason, so that you have to remember that area. And algae, you can remember chlorophyce, pheophyce, rhodophyce. Frequently I am asking I am getting questions. The way of asking questions, of course, are not wider area, but they are asking in algae members, chlorophyce. Pyrenoids are present. Pyrenoids. So if you getting the question pyrenoids, that is only for chlorophyce, particularly Spirogera. And unicellulars, multicellulars, filamentous. Ulothrix is the filamentous. Spirogera is the filamentous. Unicellular is the chlamydomonas. Chlorella is the unicellular. Chara, it is the chlorophyce. It is like plant-like algae. Colonial algae is the volvox. So that questions. Flagella is present in the chlorophyce, but it is a what kind of flagella? It is a equal flagella. But when come to the pheophyce, unequal flagella is present. Unequal flagella. But when come to the rhodophyce, there is no flagella. Flagella is completely absent. And complex sugars are present in what kind of the algae? That's you have to remember. Complex carbohydrates. And Floridian starch is present in rhodophyce. And this starch is present in the chlorophyce members. And cell wall nature, you have to remember. So that is about the most of the plant kingdom the questions are asking in this area. And of course you did learn all the questions. Many grand tests you have been writing and written so many examinations, so that you need not to worry. But I am just indicating you, just caution you while you are attempt a question answer, but you read carefully the question. Assertion reasons you have to carefully read. So they are asking what kind of the questions, that is important. Right. And also the students are getting most confused area is and also majority of the time students are coming and asking the area is plasmids, PBr 322. So dear students, you have to remember PBr 322. So the PBr 322, it is a synthetic plasmid, double strand DNA. It is a polynucleotide, not polypeptide. The polypeptide is the restriction enzymes. Wherever you find the enzymes, that is the polypeptide. Wherever you find the DNA, that is the polynucleotide. For example, if you see the DNA PBr 322, ampicillin, tetracycline, selectable markers, and the rope, it is a replication of plasmid, a protein which is essential for replication of plasmid and origin of replication. And you can see here PVU1, PST1, PAMH1 and SAL1, of course CLA1, HIND3 and EcoR1, and you can see the PVU2. Of course, whatever the questions. Here, whatever the question is asking, majority of the students are getting confused. If the alien DNA insert into the ampicillin, this will be inactivated and tetracycline is activated. So this is the thing. But what is happening here? The foreign DNA when insert into this area, this is inactivated, and these plasmid containing bacteria, if you grown in the medium of ampicillin, that is not grown. But whereas the tetracycline, it is grown. In tetracycline medium, if the bacteria are grown, this indicated what? The answer is they are the transformants. If the same bacteria or same host cells will be transformed into the ampicillin, it cannot be grown. This indicated these are recombinants. So the recombinants cannot grow. The transformants will grow. And also important thing, very very important, frequently I am getting, all recombinants are transformant, but all transformants are not recombinant. So this is the plating technique are asking. But the alien DNA, if you are inserting other than the selectable markers like Rho, like EcoR1, like CLA1, and also like HIND3, these two areas are active so that they are grown in both the areas. So that is the important. And beta galactosidase, if the enzyme is inserted into the beta galactosidase, what is happening here? The blue color should be inactivated so that recombinants are colorless, non-recombinants are blue color. But frequently the question is asking, recombinants are white color, that is also right answer. Recombinants are white color, that is also right answer. So you have to students remember. And also bioreactors, downstreaming process, not upstreaming, this is a downstreaming process. And also purification and separation and formulation, these three are the downstreaming process. And quality, marketing and also clinical trials not comes under downstreaming process, you have to remember. And sticky ends, when they are coming, if you cut into the eco R1, sticky ends will become, and phosphodiester bonds only divided and cut by the eco R1 cannot be hydrogen bonds, but they are cleaved. So these areas you have to remember. First Hindu 2, 900 and restriction enzyme from 230 steins. And what you have to remember, the scientist names also, Herbert Boyer and Stanley Cohen, that is also important. And these areas you have to remember.